what I did for my master's work at the University of New Orleans, where I looked at this, the factors that influence the spatial structure in a urban population of Anolis carolinensis in a park in New Orleans. Of course, the study organism I used is Anolis carolinensis. It is also called the green animal. It's very popular in ecology right now. And it is becoming a model species if it's not already in ecology and evolutionary biology. But there's a lot of other things that make it very interesting. It was the first reptilian genome. We have its genome completely referenced. If you want to do parental analysis, there's a whole bunch of microsatellites that can be available for it. But they have two behavioral components that are very interesting. One is a female defense polygamous system where they defend areas as opposed to just individual female or multiple females. And the males had a intersexual dimorphism where there's two morphs of males. There's heavyweights and lightweight males. Heavyweight males are males who reach the snout vent length or size of 63 millimeters and greater, and all other males stay under that, or lightweights. There are only a few that actually do get to become heavyweight males. In the study, that we should define what I'm considering spatial structure, and that's basically just the way individuals position themselves in a specific habitat in regard to other conspecifics. And there are factors that influence spatial structure, such as sex, age, size. Age and size are not necessarily synonymous. Environmental conditions, of course, have a factor in it. And mating system, whether you have one mate or two mates, also depends on how you structure yourself in a habitat. But there are some measurable behaviors that you look at to also determine this. Home ranging behavior can also tell you something about the spatial structure, territoriality, whether an individual stays in its natal home range or whether it disperses, that would be philopatry dispersal. And the relatedness factors, can also play a hint in spatial structure. In my study, we had three questions that I wanted to look at, and one of them was in regards to home range, or what is the home range of a male and a female Anolis carolinensis. And it has a hypothesis based on a sexual dimorphism in between males and males being bigger. I assumed that males would have larger home ranges than females. A second one, a more complex question and hypothesis, was how does relatedness affect the spatial structure of the male size morph, those heavyweights and lightweights? And I have a hypothesis which is partially designed off of the deer enemy a little bit, where an individual, a territorial individual, tolerates a neighboring individual because of familiarity. But I went a little bit further and kind of thought maybe relatedness played a part in that, so I wanted to investigate that as well. The last thing I wanted to look at is if there was a possibility of territorial inheritance by offspring. And once again, because of the assumption that there is believed high territoriality in Anolis carolinensis, I assume that there may be inheritance from female offspring, but there would not be inheritance towards the males, towards male offspring. My study site, like I said, is a small park in New Orleans called Washington Square Park. It's only 1 hectare in area, so it is quite small. But the characteristics of it is this wrought iron fence which completely surrounds the park itself. Animals can be found on this almost all the time. It's good for basking, and it's also good for patrolling their territory, patrolling their areas. Their main habitat is these aspidistra, broadleaf bushes that make up 39 habitats throughout the park. There's small leaf holly, which they very rarely see on there, and there's some very large old growth oak trees, which they also use for perching, but they don't travel too far up, which is actually kind of contrary to a lot of their previous ecology, because they're shown to be mid-trunk animals, and in this park they kind of are more ground animals, or bush animals. Here is an overhead of the park where you can see around the whole park itself. These are these old growth oak trees, and underneath these oak trees, surrounding the perimeter of that wrought iron fences, are these aspidistra bushes, which are the habitats. In the middle here is just broad lawn where there's a whole bunch of people congregating all the time, and we've never seen animals in that park. They'd be very highly susceptible to predation if they were there. Collections were done twice a year, once in the fall, once in the spring, from 2010 to 2015, so there was 10 collection years. We took GPSs from every individual where we caught them, took them back to the lab, took some morphological readings, and we gave them a unique tattoo. That way in subsequent sampling seasons, we could go back and see what individual we were recapturing. So with that information, the first thing we did was I made some maps in QGIS using those GPS coordinates. I first started with a map of all the different habitats in the park, gave them IDs. Then I made maps of all the seasonal captures, and then I could actually play with those a little bit, whether I segregated them by sex or size. Here's an example of just one of those maps where you can see underneath all those old growth oak trees is each one of these aspereaster habitats, 39 in total, and you can see where I actually went in there and placed each individual male or female lizard. After that, I needed to look at some genetic components, so to do that we took tail tips when we sampled them back in the lab, and we actually created genotypes for 8 microsatellite genomes. We created genotypes for 8 microsatellite markers, and in total we had 848 individuals over the 5 years that we sampled a genotype. We put those genotypes into Colony to build a pedigree, and then we put them into Spaghetti to get a lot of the relatedness data that we used for the results. So my results I'm going to break down by each question I had. The first one was, what is the home range of a male and a female green animal? Well, because of the design of the park, we had to use a design that came by Lailvaux that he worked with alligators in 2011, and because we couldn't use the middle as a corridor. And what we found, we were only able to sample, of those 848, we were only able to look at 42 females and 42 males and 39 females because we captured them more than once. Most of those individuals had 848 male colonies. And what we found was we found an average home range for a male to be a little over 1 kilometer squared, and for a female it was a little more, it was 1.5 kilometers squared. So contrary to my hypothesis, you would think that the females had a bigger home range, but when you actually do the statistical work, there wasn't a difference. So males and females don't have a difference in home range in this park. All right, the second question, like I said earlier, is a little bit more complex. It's looking at how relatedness plays a part in the spatial structure of the male morphs. The first thing I did was I took each individual and looked at the average distance that it was from every other individual in its cohort, and I made regressions on that. So if there's a strong territorial relationship going on between males, you would assume that once a male becomes bigger, smaller males will avoid it. But when I looked at the entire population together, there was no relationship. As they got bigger, it does not appear that there was any avoidance by smaller individuals. But there are the two size morphs, so I went ahead and segregated those out and looked at the lightweights themselves. Again, there was no relationship. And then I looked at the assumed territorial heavyweights, and again, there is no relationship, showing that they're being avoided the bigger they get. All right, also in that, we were wondering about neighbors, and like I said, I put in a little bit of kin theory into the deer enemy hypothesis, but it didn't stand. What I actually found was the neighbors of heavyweights were significantly less related to it than the rest of the population was. So it wasn't tolerating anybody because it was related to it at all. And also when I actually took out the confounding variable of other heavyweights that might be in bushes similar, there was nothing to look at there either. All right, so we also wanted to look at inheritance, whether females were inheriting territory from their mothers. So we constructed autocorrelograms to look for isolation by distance. Of course, isolation by distance would be an indication of phylopatry. We looked at males and females and we didn't find phylopatry in either one. So in order to have territoriality, you got to stay at home to inherit your mother's territory. We didn't see that. But we also looked at some instances where we could go back and trace every offspring to the mother and the father. And what we found in this is we were only able to look at 10 mothers, which we had her list of offspring. And what we saw was in her home range, 36% of her sons were captured, and 35% of her daughters were captured within her home range later. We also did it for males. We were able to get a few more males. We were able to get 16 males that we were able to trace their offspring. And you can see that 50% of the daughters were actually captured in the father's home range. But a very interesting thing is, if you look at sons, we were never able to capture. We were never able to capture a son within a father's home range. All right. We also were able to look at the inbreeding coefficient, which Spaghetti put out for us. And as our data set grew, and as we were able to put more and more generations in there, we actually started to see the inbreeding coefficient continually rising until it is kind of starting to plateau towards the end of the study at about 0.3. I believe this is probably a result of that 50% of females being found in the father's home range. There's probably first generation inbreed on there. So my conclusions, what did I find from this? It doesn't seem like I found a little bit of anything at all. So home ranges weren't any different. Relatedness doesn't seem to influence anything. Neighbors aren't related, and there's no evidence of inheritance. So what could be going on here? Believe it or not, it could still actually be size. When I took those maps and I segregated them out and just looked at the heavyweights in each sampling season, like I said, there's only a few heavyweights in the entire data set of 848. I started to see a qualitative pattern, where, as you can see here, the heavyweights are the dark red circles. Those heavyweights seem to have positioned themselves as far away from one another as they could, or in an individual bush. We never caught more than one heavyweight on the same day in the same bush. If you look in the top right, there's 3 heavyweights that are shown in that bush. What that was one, we caught this individual on day 1, and both of these other individuals on the second day. So it seems like once we removed this individual from his habitat, other individuals went in there and took over and took the opportunity. So there's an elephant in the room when you talk about territoriality in animals. Earlier this year, Jonathan Losos, in one of his Anolis published a paper that is hopefully being read by everybody, where it points out that we make a lot of assumptions in ecology, where we just go back and cite something from the 1930s or something from the 1980s that says these animals are territorial. The reality is they might not be. We should actually look at it a little closer rather than just cite somebody and say something is territorial. When I started my research and started looking at the results, it looked like it didn't make any sense to me until I stopped imagining them as being territorial, and then it kind of made a little bit more sense. I cannot say that they are or are not territorial. There's a lot of caveats in this that need to be looked at. But what I can assume from my urban population of animals is that it seems that a few heavyweights that make it to that threshold to become heavyweights do seem to display possible territorial behavior for a short period of time. We only caught one male in the same bush for more than one sampling season. One heavyweight male in the same bush for one sampling season. And we actually caught him for a year and a half. He seemed to have stayed in that same bush. But it seems like once they actually become big, they do display a little bit of territorial behavior. And as far as assuming that a bush to be a habitat, our bushes are about 134 cubic meters, which in the 1980s, Thomas Jensen did a study in Georgia where his territorial assessment was about 170 cubic meters. So one bush is roughly the same size as Jensen's was in the 1980s. So I feel confident saying that one of our aspen-using bushes seems to be serving as a territory for these heavyweight males. But as far as all the other females and males in that population, I do not see any instances of territoriality. So all my thanks to all of you for being here. My master's advisors in New Orleans, Dr. Lailvaux and Dr. Anthony,